Thanks for inviting me. My name is Dr. Cahuana Huaroto Maria. It's a great pleasure and honor to be here. Today I'll be talking about the article presentation. The title is Viruses and the Risk of Lung Cancer Prevention and Treatment. Introduction. Lung cancer is the most common cancer in men and the second most common among women worldwide, and it is the leading cause of cancer death in men. In addition, there are risk factors that predispose its development, such as smoking, exposure to asbestos, domestic use of biomass fuel, environmental pollution, genetics, and chronic disease. In recent years, research has been carried out on other infectious risk factors, such as viruses, that may play a role in relation to lung cancer, but this association has not yet been completely defined. On the other hand, it is important to know that COVID-19 has played a fundamental role in recent years, since it has been seen to produce an exacerbated immune reaction and lung injury that could induce tumorigenesis in the long term. Lung cancer in Europe. In 2020, in the 27 countries of Europe, there were approximately 318,327 new cases of lung cancer, of which 64% were men and 36 were women. The age range with the highest cases was between 45 and 69 years, and a total of 257,293 died from this cause, where 66% were men and 34% were women. The three countries with the highest incidence of cases were Hungary, followed by Ireland and Belgium, while the countries with the highest incidence of death were Hungary, Poland, and Croatia. Viruses and their relationship with lung cancer. According to clinical studies and several meta-analyses, they suggest that the main oncogenic infectious agents associated with lung malignancies are Epstein-Barr virus, human papillomavirus, hepatitis B and C. Likewise, smoking has a potential role in increasing the susceptibility of lung tissue to viral infection, in addition to affecting the function of oncoproteins. It is noted that tobacco smoke alters the immune response, contributing to viral pathogenesis and causing deterioration in lung function. Human papillomavirus is a double-strand DNA virus. It is associated with the development of several types of cancers in humans. It has more than 200 serotypes, of which types 16 and 18 are responsible for the majority of cancers. Its genome encodes various oncogenic proteins that act as factors that stimulate tumor development by promoting the cell cycle and inhibiting apoptosis. A recent systematic review and meta-analysis including 78 studies with data from 9,385 patients examined the presence of this virus infection in lung cancer. Human papillomavirus was detected in 1,268 cases. Additionally, 6.1% were serotype 16 and 3.1% were serotype 18. Another case control study identified human papillomavirus DNA in 53% of cases and 25% of controls. Human papillomavirus 16 was the most commonly identified virus type. This demonstrates a significant association between this virus and lung cancer. Epstein-Barr virus. This agent is associated with a particular subtype of primary lung cancer called lymphoepithelioma-like lung carcinoma, accounting less than 1% of cancer that frequently affects younger, non-smokers, and the Asian population. On the other hand, this virus has also been found in non-small cell lung cancer in patients from the USA and in isolated air condensate samples from patients in Italy. Epstein-Barr virus goes through two different stages that complete its life cycle, a latency and lytic cycle. There are four stages of latency. In latency phase 2, nasopharyngeal carcinoma and lung cancer are observed, where proteins such as EVNA1, latent membrane protein 1, 2I, 2B, EVER, and BAR are expressed. Several studies have reported the detection of Epstein-Barr virus DNA in immunosuppressed intensive care unit patients with chronic lung disease, as well as in COVID-19 patients with severe lymphopenia. Current data suggests that Epstein-Barr virus latency could facilitate the progression from chronic lung disease to lung cancer and vice versa. Human immunodeficiency virus. People living with this virus are more likely to develop lung cancer because their immune status has deteriorated and chronic inflammation is established despite antiretroviral treatment. A core study showed that the risk of this type of cancer was related to not recovering CD4 count of approximately 500 cells millimeters. Antiretroviral therapy was initiated. In addition, other authors have also found an association with lung cancer infections, for example, pneumocystis jirovecii pneumonia and pulmonary tuberculosis, more than 10 years after their appearance, acquired immunodeficiency syndrome. Other research expands the use of gene expression for the early detection of lung cancer in patients with this virus, where the overexpression of genes such as promining 1, AP2 alpha transcription factor, and sinus oculis homeobox homolog 1 was found, among others. This finding opens avenues for the use of genetic biomarkers in timely diagnosis and follow-up in treatment. Cytomegalovirus is a double-strand DNA herpes virus. This remains in the latency stage for long periods and can be reactivated intermittently through life. This promotes chronic inflammation, genomic mutability, and immunosuppression in the host, which can lead to the initiation of cancer. In a recently published series of 144 consecutive cases of malignant pleural mesothelioma, anti-cytomegalovirus IgG was detected in 51% of the cases, and also cytomegalovirus DNA in 48% of the tumors. However, there are no reports indicate a direct relationship between cytomegalovirus and lung cancer, although cytomegalovirus have been found in a variety of tumors. Measles virus. This virus shows tropins for the oropharyngeal epithelium. In 2002, Sayombardi et al. Detected measles virus P53 and P53 induced ring H2 antigens in 54 of 65 non-small cell lung cancer cases. Jackson sheep retrovirus and John Cunningham virus. GS retrovirus causes lung cancer in sheep. In humans, GS retrovirus infects cells through the cell surface hyaluronidase 2 receptor. One study suggests a possible link between GS retrovirus and human lung adenocarcinomas. On the other hand, JC virus has been detected in several types of human tumors, suggesting that GC virus has the ability to infect a wide range of cell types. Prevention and treatment of oncogenic viruses. Prevention of infection by oncogenic viruses is complicated science, with the exception of human papillomavirus. There are no approved vaccines for the viral agents described above. However, there is no data to support a decreased incidence or risk of lung cancer in human papillomavirus vaccine population. On the other hand, strategies have been proposed to avoid viral to block or reduce oncogenic transformation. For example, in Epstein-Barr virus infection, smokers are recommended to keep as primary prevention measure for upper respiratory tract carcinomas. Additionally, there are currently several phase 3 clinical trials evaluating drugs and vaccines. Another phase 3 trials of biological therapy where multivirus-specific T cells are used in patients undergoing allogeneic stem therapy. This new therapy could provide a treatment for patients infected with oncogenic viruses in the future. Conclusion. The role of viruses in the development of lung cancer is still unclear, but it is supported by epidemiological data on infection in tumor cells, which is why more research is still needed. Finally, smoking may contribute to viral oncogenesis by producing local immunosuppression and increasing viral pathogenesis. Thanks for your attention.